Have you ever paused to think about the intricate, almost unbelievable engineering that goes into creating and sustaining a new human life? Right from the very beginning, inside the womb.
It really is something else. The complexity, the orchestration.
Yeah.
It's truly biological artistry.
It feels like science fiction sometimes. So today, we're taking a deep dive into some of the most vital, yet often overlooked players in this whole process. The placenta, the fetal membranes, and the umbilical cord.
The unsung heroes, definitely. Critical connectors bridging two lives.
Exactly. And they make all that incredible development possible. Our mission here, using a whole stack of detailed sources, is to unpack these really complex biological systems.
Reveal the surprising facts, the crucial insights, understand why they're so essential.
Okay, let's unpack this incredible trio that makes life in the womb possible.
It's a truly foundational area of human biology, often taken for granted, maybe because these structures are temporary.
It's probably true.
But understanding their roles is absolutely critical for appreciating, how a healthy pregnancy progresses and how the fetus actually thrives.
I couldn't agree more. Let's begin with the placenta. An organ so unique it defies belief. It's only found in eutherian mammals like us.
That's right.
And the human placenta has some very specific features. It's discoid, so disc-shaped.
Yeah.
It's hemochorial, which refers to how the fetal tissue makes direct contact with the mother's blood.
Direct contact, yeah.
And it's deciduate, meaning a bit of the maternal uterine lining actually sheds at birth along with the placenta.
And here's where one of the most, frankly, mind-blowing aspects comes in. Think about it.
Okay.
The human body is incredibly good at recognizing and getting rid of anything it sees as foreign. That's our immune system, right? Protecting us.
Right, rejection, like transplants.
Exactly. Yet for 9 months, a mother's body completely accepts this tissue that's, genetically speaking, half foreign.
Yeah.
It's got paternal genes. This isn't just fascinating. It's like one of biology's biggest mysteries. A masterclass in immune tolerance. Scientists are still trying to figure it out, hoping maybe we can replicate it for things like organ transplantation.
That truly is a huge aha moment. And it's not like it's just there from day one, fully formed, is it?
Oh, no, not at all. It has this remarkable journey of development.
It's got a dual origin, I read.
Oh.
Part fetal, part maternal.
Precisely. Primarily fetal, from a part of the early embryo sac called the chorion frondosum, and then a maternal part from the uterine lining, the decidua bassalis.
And it forms incredibly quickly.
Amazingly fast. Starts with tiny spaces in cells. Around day 11, you get these early structures, stem villi. By day 13, and by day 21, there's already a complete blood vessel system connected to the embryo.
Wow, day 21.
Yeah. And at the same time, those tiny spaces, the lacunae, they expand and merge. By the 3rd or 4th week, they form what becomes the crucial intervillous space.
That's where the maternal blood will eventually flow.
Exactly. That's the pool, basically. Then the placenta itself really grows dramatically, becoming a distinct organ between the 6th and 12th weeks.
So quite early on, really.
Oh, yeah. It keeps growing in thickness and circumference until about 16 weeks, and then mostly just expands outward circumferentially until term.
So once this incredible organ is fully developed, say, at term, what does it look like?
Okay, so when it's delivered, it's typically a circular disc,
Yep.
about 15-20 centimeters across, roughly dinner plate size, like you said,
Right.
and maybe 3 centimeters thick in the middle, thinning out at the edges, weighs about 500 grams, so just over a pound.
And that's about 1/6 the baby's weight, roughly.
Yeah, roughly 1.6 ratio, and it takes up quite a bit of space, about 30% of the uterine wall.
Okay, so looking at the side facing the baby, the fetal surface.
Right, that side is smooth, kind of glistening. It's covered by that thin, transparent membrane, the amnion.
Oh, okay.
And the umbilical cord usually attaches near the center, and you can clearly see the baby's blood vessels radiating out from where the cord joins. It's quite striking.
And you mentioned most of it is fetal tissue.
That's right. About 4/5, 80% of the placenta is actually a fetal origin. It's the baby's organ, primarily.
And then you flip it over the maternal side.
Totally different look. It's rough, spongy, and a kind of dull red color because of all the maternal blood.
Makes sense.
And this surface is divided into these 15 to 20 somewhat convex, bumpy areas. Those are called lobes or cotyledons, separated by little grooves, which are actually folds of the decidua.
I've seen pictures. Sometimes they have little white spots.
Yeah, you might see small grayish or whitish spots. That's usually just calcium deposition. Pretty common, generally nothing to worry about clinically.
Gotcha. And internally, what's the structure like inside?
Well, internally, it's cleverly arranged. You've basically got two main plates. There's the chorionic plate on the inside, the fetal side where the cord attaches,
Okay.
and then the basal plate on the outside, the maternal side, attached to the uterus.
And between them is that space.
Exactly, the intervillous space we talked about.
Yeah.
It's basically filled with slowly flowing maternal blood like a shallow lake. And suspended in that lake are all the stem villi and their branches, like trees dipping into water.
And these stem villi,
Yeah.
they're the real workhorses, right? Doing the exchange.
Absolutely. They sprout from the chorionic plate, anchored down onto the basal plate, and branch out extensively, creating this huge surface area for exchange.
How huge are we talking?
Get this. The total exchange surface area is estimated at 10 to 14 square meters.
Wow, that's like the floor area of a small room.
It is. And the fetal capillary system within those villi, if you laid it out end to end, it'd be almost 50 kilometers long.
50 kilometers.
Hmm.
Incredible.
And at the microscopic level, where the real action happens, the outer layers of these villi, especially near term, become incredibly thin in spots, forming what's called a vasculo-syncytial membrane.
How thin are we talking?
We're talking just a few thousandths of a millimeter thick sometimes, thinner than spider silk. This ultra-thin barrier is absolutely key for efficient gas exchange oxygen in, CO2 out.
Which brings us nicely to the dynamic exchange itself, the placental circulation. Two separate systems working together.
Exactly. Two entirely independent yet very closely intertwined circulatory systems. It's really quite elegant.
So first there's the utero-placental circulation. That's the maternal side, right?
That's mom's contribution. The mature placenta holds about 500 milliliters of her blood in total, with about 150 mils specifically in that intervillous space at any given moment.
And the flow rate?
Astonishing. About 500 to 600 milliliters per minute, which means the blood in that intervillous space gets completely replaced about 3 to 4 times every single minute.
Whoa. Constant turnover. Where does it come from?
It arrives through about 120 to 200 specialized arteries in the uterus called spiral arteries. They pierce through the basal plate into the intervillous space.
Now here's where it gets really interesting, I read.
Yeah.
Something about fetal cells changing these arteries.
Yes, this is truly ingenious. Fetal cells, specifically trophoblasts, actually invade these maternal spiral arteries early in pregnancy.
Invade?
Yeah, they migrate into the artery walls, replace the mother's own endothelial cells, and even destroy some of the smooth muscle layers.
Why would they do that?
To convert these normally narrow muscular arteries into wide, floppy, low-resistance vessels, like turning narrow pipes into wide funnels.
Oh. So it reduces the pressure.
Exactly. It significantly reduces the blood pressure as it squirts into the intervillous space down to maybe 70, 80 millimeter Hg and ensures a steady, high volume of blood flow without damaging the delicate villi.
That's critical.
Absolutely critical. And what's fascinating, or perhaps concerning, is that defects in this trophoblast invasion process are known to be linked to serious pregnancy complications, things like preeclampsia or fetal growth restriction. It really highlights how vital this early remodeling is.
Wow. Okay, so the blood comes in via these remodeled arteries. How does it get out?
Venous drainage happens through uterine veins, which also just randomly pierce the basal plate. Blood flows out through those.
And inside that space, the intervillous space, how does the blood move? Does it just pool there?
Well, it enters in these sort of pulsing jets from the spiral arteries, but the dense network of villi disperses these jets, slowing the flow and helping mix the blood for optimal exchange.
Like baffles in a tank?
Sort of, yeah. And even gentle pressure from uterine contractions and the subtle pulsation of the villi themselves probably helps circulate and mix the blood.
You mentioned tiny bits of the placenta breaking off.
Yeah.
Syncytial sprouts.
That's right. Tiny fragments of the outer syncytial layer are shed into the maternal bloodstream maybe 100,000 in 24 hours. They circulate and are then naturally cleared, mostly by the mother's lungs. Doesn't seem to cause any harm.
And how does all that blood not clot in there?
Good question. The trophoblast cells produce fibrinolytic enzymes, natural blood thinners basically, which helps prevent clotting within the intervillous space.
Okay, that covers the maternal side. What about the fetal side? The fetal-placental circulation?
Right, so this involves the umbilical cord vessels. There are 2 umbilical arteries.
Wait, arteries usually carry oxygenated blood, right?
Usually, yes, in systemic circulation, but here it's reversed, like in the pulmonary circuit. The 2 umbilical arteries carry deoxygenated waste-rich blood from the fetus to the placenta.
Okay. Got it. from the fetus.
And then there's the single umbilical vein, which carries the freshly oxygenated, nutrient-rich blood from the placenta back to the fetus.
2 arteries away, 1 vein back.
And the arrangement within the villi is super efficient. It's set up for countercurrent flow.
Countercurrent, meaning?
Meaning the maternal blood in the intervillous space flows past the fetal capillaries in the villi, but in opposite directions. This maximizes the concentration gradient all along the exchange surface, making the transfer of oxygen, nutrients, and waste incredibly efficient.
Clever design.
Very clever. And this whole fetal circulation loop is driven by the fetal heart pumping. The flow rate on the fetal side is about 400 milliliters per minute.
Right, now let's talk about this placental barrier. It sounds like a wall, but you're saying it's more like a selective gate.
Exactly. It's often called a barrier, but that's a bit misleading. It is a partition between the fetal and maternal circulation, absolutely, but it's not perfectly impenetrable.
Because cells can cross sometimes.
Precisely. Tiny amounts of fetal cells can get into the mother's circulation, and maternal cells can sometimes cross into the fetus. So it's more like a very, very tightly controlled border crossing than a solid wall.
And it changes during pregnancy. You mentioned it gets thinner.
Yes. In early pregnancy, it's relatively thick, maybe 0.025 millimeters. It has several layers. The outer syncytiotrophoblasts, then an inner layer of cytotrophoblast cells, a basement membrane, some connective tissue called stroma, and finally the wall of the fetal capillary, the endothelium.
Okay, several layers.
But as pregnancy progresses towards term and the fetus needs more and more oxygen and nutrients, this barrier thins out dramatically in certain areas. Those vasculosyncytial membranes we talked about can get down to just 0.00002 millimeters thick.
Wow, incredibly thin.
That thinning is a critical adaptation for boosting the efficiency of gas exchange right when the baby needs it most.
So how do things actually get across? You've got gases, nutrients, waste. It must use different methods, right?
Absolutely. It's a whole toolkit of transport mechanisms. For small stuff like oxygen, CO2, water, electrolytes, it's mostly simple diffusion. They just move down their concentration gradient. Easy peasy.
Okay.
Then for glucose, the baby's main fuel, that uses facilitated diffusion. Special transporter proteins like GLUT1 act like specific doorways to help glucose cross much faster than it would by simple diffusion alone.
Like a revolving door for glucose.
Kind of, yeah. Then for things the baby really needs to concentrate, like amino acids for building proteins or ions like calcium, phosphorus, iron, and also water-soluble vitamins, the placenta uses active transport.
Meaning it uses energy.
Exactly. It actively pumps these substances across, even against a concentration gradient. It can pull these vital nutrients from the mother's blood, even if her levels aren't super high, ensuring the baby gets priority.
That makes sense. What about bigger molecules, like proteins, antibodies?
For larger molecules like proteins, including important antibodies like IgG, which give the baby passive immunity, the placenta uses processes called endocytosis and exocytosis. Basically, it engulfs the molecule on one side and releases it on the other.
Like Pac-Man.
A very sophisticated Pac-Man, yes. There are other mechanisms, too, like bulk flow, where water movement carries solutes along, and as we mentioned, even tiny leaks that allow cells through occasionally.
So what specifically crosses? We've mentioned nutrients.
Right. Glucose is key for energy. Lipids for building fetal fat come from both the mother and synthesis in the placenta fetus. Amino acids are actively transported. Water, electrolytes, iron, vitamins all cross.
And waste goes the other way.
Yep. Waste products like urea, uric acid, creatinine just diffuse easily from the fetal blood to the maternal blood for the mother's kidneys to handle.
What about hormones? Do they cross?
Some do, but often slowly or poorly. Insulin, adrenal steroids, thyroid hormones. They cross, but not readily. And some, interestingly, like parathormone and calcitonin involved in calcium regulation, don't seem to cross at all. The fetus largely manages its own hormonal environment for those.
But you said the barrier isn't perfect. Infections, drugs.
Yes, and this is a really critical point. While it generally holds back large molecules, many infections and drugs can definitely cross.
Like what kind of infection?
Viruses are a big concern. Rubella, chickenpox, measles, mumps, polio, HIV, Zika. Bacteria like the one causing syphilis, Treponema pallidum, tuberculosis. Protozoa like Toxoplasma gondii from cat litter or undercooked meat. And the malaria parasite.
That's sobering.
It is, and it underscores the importance of vaccinations and avoiding exposure during pregnancy. And then there are drugs.
What about them?
The reality is almost any drug taken by the mother has the potential to cross the placenta to some extent. Some easily, some less so, but the potential is there.
That's why doctors are so cautious about medications during pregnancy.
Absolutely. The risks versus benefits always have to be carefully weighed because the fetus could be exposed.
Okay, so beyond this incredible exchange function, the placenta does other things too. Enzymes.
Yes, it has significant enzymatic functions. For example, it produces an enzyme called diamine oxidase, which helps break down histamine and other pressuramines, potentially regulating blood pressure. It also makes oxytocinase, which can neutralize oxytocin, maybe helping prevent premature contractions.
Interesting. And the big one we touched on earlier, the immunological function. How does it avoid rejection?
Ah, yes, the great mystery. Why isn't this half-foreign tissue rejected like a mismatched organ transplant?
Yeah, how does it work? What are the theories?
Well, it seems to be a combination of factors. One idea is that placental hormones and proteins like HCG, progesterone, and specific proteins like SP1, EPF, PPPA actually have immunosuppressive effects locally, dampening the mother's immune response right at the interface.
So calming the immune system down nearby.
Sort of. And perhaps even more importantly, the main placental cells in contact with maternal blood, the syncytia trophoblast, do not express the typical HLA class 1 or class 2 molecules that the mother's immune system usually recognizes as foreign.
They're immunologically invisible.
Largely, yes. Makes them very hard for the maternal immune cells, like T cells, to see and attack. Other placental cells, the extravillous trophoblasts that invade the uterus, express only a specific non-classical type of HLA, HLA-G, which might actually interact with maternal immune cells to promote tolerance.
Fascinating.
There's also evidence that the mother's overall immune system shifts during pregnancy. It seems to move away from cell-mediated immunity, the kind involved in rejection, toward humoral or antibody-based immunity.
A system-wide shift.
Possibly. Plus, there are specialized immune cells in the uterine lining, the decidua, particularly decidual natural killer, NK cells. They seem to interact directly with trophoblast cells in a way that helps regulate invasion and might promote tolerance rather than rejection.
So NK cells helping, not hurting.
In this context, yes, it seems so. And finally, one theory suggests the mother might produce specific blocking antibodies against certain paternal antigens, maybe TLX antigens, which could coat the fetal cells and protect them from attack by other immune cells. It's likely a complex interplay of all these mechanisms.
Truly remarkable biological diplomacy.
Absolutely. And like any hardworking organ, the placenta does show signs of aging as it approaches term. Natural degenerative changes the syncytium and thins, some cells disappear, some vessels might get blocked off. It's fulfilled its primary role.
Okay, let's shift focus slightly to the fetal membranes, the protective bubble around the baby.
Right, the amniotic sac. It has two main layers. The outer one is the chorion, thicker, a bit fragile, looks kind of shaggy. It's the remnant of the early chorion leaf.
And the inner layer.
That's the amnion. It's smooth, shiny, tough, and it's directly in contact with the amniotic fluid. Importantly, it doesn't have its own blood supply or nerve supply.
And the sac just grows with the baby.
Essentially, yes. The amniotic cavity starts small, but enlarges dramatically as fluid accumulates, eventually surrounding the fetus completely, providing that protective, fluid-filled environment.
And the amniotic fluid itself, where does it come from? Is it just baby pee?
Well, fetal urine becomes a major contributor later on, but the origin is actually quite complex and still not fully understood.
Yeah.
It's likely a mix. Early on, probably mostly a transudate fluid filtering across from the mother's serum and maybe the fetal circulation. Secretion from the amniotic epithelium itself likely plays a role. Fluid transfer across fetal skin is important early, before the skin keratinizes.
And then later?
Later, as the fetal kidneys start working, fetal urine becomes a very significant source, yes. And fluid secreted by the fetal lungs also adds to the volume, so it's a dynamic mix from multiple sources.
And you mentioned its dynamism earlier. The turnover rate.
That's one of the most amazing things. The water in the amniotic fluid is completely changed and replaced every 3 hours or so. It's incredibly dynamic.
Every 3 hours?
Yeah, mostly through fetal swallowing and fetal urination. At term, the fetus swallows a huge amount, maybe 500 to 1,000 milliliters a day.
Wow.
And they produce a similar amount of urine, 400 to 1,200 milliliters daily at term. This constant cycling is key to maintaining the fluid volume and composition.
And the total volume changes over pregnancy.
It does. It increases steadily. You might have only 50 milliliter at 12 weeks, but it reaches around a liter, maybe 1,000 milliliter by 36, 38 weeks.
A whole liter.
Yeah. Then, interestingly, it tends to decrease slightly as the pregnancy goes towards term, maybe 600, 800 milliliter, and can decrease further if the pregnancy goes significantly post-term.
And the appearance of the fluid can tell you things.
Yes. Clinically, very important. Normally, it's clear or colorless in early pregnancy, then gets a pale straw color near term, maybe with little white flecks of vernix from the baby's skin.
But abnormal colors mean trouble.
They can. Meconium staining, that's the baby's first stool, makes it green, and that can be a sign of fetal distress. A golden yellow color might indicate Rh incompatibility due to bilirubin from excessive breakdown of fetal red blood cells. Greenish yellow or saffron can be seen in post-maturity.
Any others?
Dark-colored, like port wine, might suggest a concealed placental abruption, which is serious bleeding. And a dark brown, like tobacco juice, is unfortunately often a sign of intrauterine fetal death, IUD. So color is a key diagnostic clue.
Wow. What are the main jobs of this fluid besides just being there?
Oh, it has many crucial functions. During pregnancy, it's a fantastic shock absorber, protecting the fetus from bumps and jolts.
Like internal bubble wrap.
Exactly. It also maintains an even, stable temperature around the fetus. Critically, it allows space for the fetus to grow symmetrically and move freely.
Movement is important for development, right? Muscles, joints.
Absolutely. Without that free movement in the fluid, you'd get contractures and restricted growth. It also prevents parts of the fetus from adhering to the amnion.
And during labor. Does it still have a role?
Yes, several important roles. The intact sac of fluid, the bag of waters, forms a hydrostatic wedge that helps gently dilate the cervix before the baby's head engages.
Ah, using fluid pressure.
Right. It also cushions the umbilical cord during contractions, preventing compression that could cut off the baby's oxygen supply. It helps maintain placental circulation during contractions, too. And when the membranes rupture, it flushes the birth canal, providing some aseptic and even bactericidal protection against infection moving upwards.
So it's useful right up to the end.
Definitely. And clinically, measuring the amount of fluid, often using the amniotic fluid index, or AFI, on ultrasound is a key part of assessing fetal well-being. Too much fluid, polyhydramnios, or too little oligohydramnios can indicate underlying problems.
Okay, that leaves the third part of our trio, the umbilical cord.
The lifeline.
The direct connection. It develops from what's called the connective stalk or body stalk, which initially links the tiny embryonic disc to the outer sac, the chorion.
And it moves. Yeah, as the abdomen develops and closes, the attachment point shifts towards the center of the baby's belly, usually getting there by about the 4th month.
And inside the cord, what protects those vital vessels?
That's the job of Wharton's jelly. It's this unique, gelatinous, mucoid connective tissue, rich in mucopolysaccharides. It surrounds the blood vessels completely.
Why is it so important?
It provides incredible protection. It's resistant to compression and torsion, preventing the umbilical vessels from kinking or getting squeezed shut, which would obviously be catastrophic for the baby. It's like natural protective padding.
And the vessels themselves. We mentioned 2 arteries, 1 vein.
That's the standard setup, yes. It actually starts with 2 arteries and 2 veins, but one of the veins usually degenerates and disappears quite early on.
Leaving the 2 arteries carrying the impure blood away from the fetus.
Correct. Deoxygenated blood and waste products going to the placenta.
And the 1 vein carrying the oxygenated, nutrient-rich blood back to the fetus.
Exactly. Oxygen and goodies coming back in.
And finding only 1 artery is significant.
Yes, finding a single umbilical artery, or SUA, is a relatively common anomaly, maybe seen in up to 1% of pregnancies. While many babies with SUA are perfectly healthy, it is associated with an increased risk of other congenital abnormalities, particularly cardiac, renal, or gastrointestinal issues, so it prompts a closer look with ultrasound.
Good to know. What about the cord itself? Length, knots.
Average length is around 40 centimeters, maybe 50, but there's huge variation. Anywhere from 30 to 100 centimeters isn't uncommon. Diameter is about 1.5 centimeters.
Long cords could get tangled.
They can. You often see false knots, which aren't true knots, but just kinks or bulges of the vessels within the Wharton's jelly. They usually don't cause problems. Actual true knots, where the cord literally ties itself in a knot, are much rarer, maybe 1% of the time. They can tighten during labor or delivery and restrict blood flow, which is a concern.
Does it twist?
Yes, the cord typically has a characteristic spiral twist, usually twisting from left to right. The reason for the twist isn't fully clear, but it might add strength and resist kinking.
And those umbilical arteries.
Phew.
You mentioned something unique about their structure.
Ah, yes. Unlike most arteries, they lack an internal elastic lamina, that stretchy layer. But instead, they have remarkably well-developed muscular coat, thick muscle walls.
Why is that important?
It allows them to constrict powerfully and rapidly due to spasm shortly after birth, triggered by the change in temperature and handling. This helps clamp down blood flow very effectively, minimizing blood loss from the baby before the cord is formally clamped and cut.
Nature's own clamp.
Pretty much.
And where the cord attaches to the placenta can vary too.
Yes, the insertion point isn't always dead center. It can be eccentric, off-center, central, marginal, right at the edge, sometimes called a battledore placenta, or even velamentis.
Velamentis, what's that?
That's where the umbilical vessels actually separate and run through the fetal membranes before they reach the placental disc itself. They are unprotected by Wharton's jelly for a stretch.
That sounds risky.
It can be, especially during labor if those membranes rupture near the vessels or if the vessels run across the cervix, thus a previa. It requires careful monitoring.
Wow. Okay, so we've journeyed through the placenta, the membranes, the cord, this incredible support system. When you pull it all together, what's the big takeaway?
I think the big takeaway is just the sheer elegance and efficiency of it all. These are temporary structures built entirely from scratch